This last talk is really like a conference talk. You've seen what precision medicine is. You've seen the tools we use. You've seen the algorithms behind these tools, and you've seen the outcomes that can be had. What's missing so far is like hard science, and so I mean we would be remiss if we didn't give you some of that hard science. So here it is. I'm not looking forward to this, and I hope you enjoy it. Right after me comes the delightful Gail King, and she'll be talking about how to use this and customize what we say in our protocols with what is available in your clinics and what you are currently practicing. So this is kind of, we talk about models, right, briefly, but like how is a model generated and how many years and studies does it take to create something like this? I'm not going to go into breast cancer. You guys know what it is. You know triple negative. You know ductal lobular. You understand histology. You understand that there is 89% survival rate, 5-year survival rate for breast cancer, and you know that that can be as high as 99% if it's caught in stage 1 and 2. So we're not going to go into the epidemiology and facts about breast cancer. It is the #1 cancer affecting women. We know this. We're just going to go into intro to in vitro diagnostics. What are they? You'll notice at the back of our box we have an IVD stamp. What is an in vitro diagnostic? Then we're going to talk about breast cancer metabolomics. We're going to introduce a type of profiling that we developed called database-assisted mass spec, globally optimized targeted mass spec, or DGOT mass spec. We'll talk about how we applied different types of metabolomics for early detection of BC. It's a follow-up to a first study we'll talk about. And then we're going to talk about metformin for breast cancer. And it's a pooled analysis of 2 randomized controlled trials and some of the impacts we saw. So these are all mass spec metabolomics-based studies we're going to cover. So what is an in vitro diagnostic, right? So these are lab-developed tests. They are collected and data is generated in a single facility. So we would not be an LDT if we did part of our processing in this building, and then we took that and analyzed it on a mass spec in the building next door. Everything has to be done under regulation in one building. This is an in vitro diagnostic, is a laboratory-developed test. They're affectionately known as home brews, and they are a mechanism by which researchers like me can respond dynamically to emerging health threats or to produce tests that are going to be conducted in a laboratory setting, even if disseminated to individuals, that can look at diagnoses much quicker, cheaper, with more accuracy, and if possible, with a less invasive detection method. So there are some key features of these, right? They analyze biological samples. They give some critical health information beyond what's available with traditional testing. They are conducted, again, in vitro, obviously, as the name suggests. And they lead to things such as more timely diagnoses when they're used correctly, disease monitoring, screening programs, and of course, personalized medicine. What you see on the right is a Cobras autoanalyzer for molecular chemistries. And on the right is a point-of-care test. These are both in vitro diagnostics. I usually work on the machine side of things, but I have developed these lateral flow assays as well and published on them many times. They're boring, but these are technically in vitro diagnostics as well. Things like pregnancy tests or, in the case of my own work, neutralizing antibodies for COVID vaccination. Why don't we have these? Like, what's going on? Like, why are these sort of not prevalent? You know, we have these testing services, but they're not really rooted in science. The serious scientists, at least, are not usually at these companies. And what are the bottlenecks in bringing these to the market, right? Number one, we have 4 of these domains that I postulated in my doctoral dissertation. And so this is unpublished. So this is regulatory and scientific, some things about study design, pre-analytical considerations, and post-analytical considerations. So this is how I bin them. And one thing is enhanced guidance from government entities. So the FDA just made a new ruling, which will, in 5 years' time, make tests like us subject to more scrutiny and more validation, depending on the marketing claims we make. This has been absolutely, mocked in the science and literature because this is really going to, I think, cripple how quickly scientists can respond to things like emerging health threats, which are the most important, right? So we do routine health monitoring, but there will be greater standardization across the field, which is a good thing. We want that. That holds us all to some sort of standard and also allows for translation between results. There's also, we need more incentives for translational work. Like scientists like Alex and myself, we're not incentivized to do translational work. It takes a lot of time and it results in one paper. Or we can sequence a bunch of gut bacteria and I can run some mass spec. We get a ton of data, we publish it, we go on to the next thing. We are incentivized to continuously discover. We are stuck in a discovery loop, and that needs to stop so that the translational work can begin. So that's what we always talk about as far as the parallel institution that we're building. And then improve collaboration with regulatory bodies. So I created this point of care test. I was the biostatistician for that team. I'm not an immunologist. I know nothing about that. And we got this approved by the FDA for the claim we're marketing. But we did that because from day one, when we started thinking about, hey, we want to make an antibody test, but we want to specifically look at neutralizing antibodies, and how can we create this lateral flow paper with binding pockets specific to that neutralizing antibody, we started to engage the FDA at the beginning. And this was through a company, Axum Biotech, which is in San Diego. And that's not really the thing. There's no FDA here, right? Like, we need FDA to be more involved. If we have to go through them, then we would like them to be more involved from inception. Study design. Lots and lots and lots of times we have poor study design, whereby like we look at 20 people and then we say, okay, here's a biomarker. That's not valid. What we really need to do is look at study design and increase sample size. Also increase sample size across locations. There are biomarkers for geography where you can do these studies and figure out if somebody's from the East Coast or the West Coast just by testing their blood and looking at metabolites, right? How do we correct for those? We need to do multi-site studies. We need to have sufficiently large sample sizes in these studies whereby we're not doing internal permutation on the statistics anymore. We're able to do proper training and testing sets that respect power, that respect our chance of reducing type 2 error, right? We want to make sure that we're at 80% power, and these studies are powered for the inferences they're making. Oftentimes, they're not. Also, sometimes there's no clinical utility or parallelization, so they'll do, they'll do a cancer screen for breast cancer. I'm guilty of this. We'll see. But then we won't look at like imaging techniques. We won't parallel our biomarker panel against the gold standard, the standard of care that is currently used, to see how much better it is. So we'll produce this thing and we'll say, okay, it can detect stage 1 cancer, but what about advanced imaging techniques? More than likely, we would have beat out advanced imaging techniques because a tumor needs to be sufficiently large to be imaged, and we're looking at very nascent chemical signatures that aren't apparent yet on a visible tumor. Or we'll come up with like a test for valley fever, which we did, and we patented it. It's really prevalent in Arizona, valley fever, coccidioidomycosis. But we didn't include in our study design people with community-acquired pneumonia, which is a confounding variable. When somebody presents to the physician with symptoms of valley fever, the doctor is not considering, now is this person suffering from valley fever or are they healthy? Obviously, they're suffering from something, right? So just having a study design where it's like control healthy, that doesn't serve anything. You've just produced biomarkers that differentiate between valley fever, of active infection, and people who have no evidence of disease. What's really important is, how about community-acquired pneumonia, bacterial pneumonia? What about lung cancer? Oftentimes, valley fever is misdiagnosed as lung cancer. The tool, the utility, lies in giving the physician a biomarker panel capable of an accurate differential, not differentiating between, is this person healthy or are they diseased? Obviously, they have something. It could be a flu, right? Valley fever manifests in various ways. So we patented it, and it's absolutely useless. Enhanced use of non-invasive samples, right? We want to take blood, and we need more validation on non-invasive or minimally invasive samples like our blood spot, right? Do I want to assess your neurocognitive domain by using cerebrospinal fluid? Sure I do, but there's no at-home spinal tap maybe in the future, but we need to invest in these non-invasive sample collection methods that enable remote patient monitoring. And then pre-analytical considerations. Oftentimes, a lot of labs are sloppy, right? We have studies where I've done on urine profiles, and they change through time with respect to the temperature they're stored at and with respect to the preservatives they're used. Sometimes we use boric acid or thymol. And we did this study and published it, and we saw that urine samples we're getting from different labs from the same person are completely different. So standardize how they're handled, standardize how they're prepared. We need to have more guidance, and this is going to come from bodies like the MSACL, the Mass Spectrometry Association for Clinical Laboratories, on how we're going to prep these samples. There is ways to get short-chain fatty acids 1,000 ways. There's 1,000 ways to get a short-chain fatty acid panel. Slight differences in mass spectrometry conditions, slight differences in sample preparation, and they may or may not return similar results, but we need to come together as a science and create these formalized positions and saying this is how we're going to measure short-chain fatty acids. This is how we're going to measure aqueous metabolites. And then enhance reporting of quantitative results. Ultimately, we want to get to a point where everything is benchmarked against an internal standard. We have calibration curves, and we're giving truly quantitative interpretation. Right now, when we publish a study with our GCMS, it's relative abundance. It cannot be compared to another study. We need to start producing things in absolute concentrations. We can use it for single sample profiling and health monitoring, especially with that same individual on the same instrument through time. But we can't compare our results one-to-one with published data. So we usually use correction factors to adjust for batch effects. So this needs to come up. Same thing happens with post-analytical considerations. Some people use different softwares for processing those peaks. Some people use a completely different software for identifying which peaks are present at which retention times. That will change the quantitation of that metabolite. We need more curation of biomarker databases. There are tons of databases that you'll pull up and you say, I want the spectra of 4-hydroxybenzoic acid. And you won't have a confirmed spectra. So there's nobody who's run that sample with a standard, put up the spectra for other researchers to benefit from. We need to incentivize this so that as researchers create spectra, confirmed spectra from standards, they're depositing this and we can all sort of benefit. And then enhanced accessibility and scalability of tests. This will come later. These are things like the miniaturized mass spec, as I talked about. This device here is a lab on chip. You may have heard about lab on chip devices. They are able to process and compute roughly half a dozen analytes at once. They're high throughput and they're incredibly sensitive. But we need to bring this forward, right? These are the sort of overarching goals ultimately of Theriome. So first thing we're going to talk about is my first ever publications. We're going back to 2019, which is the year I feel comfortable. I don't really report on any studies prior to 2019. I don't include them in my research because it's outdated. But here we have a good example of a paper that we published in the JCB. And we're looking at breast cancer detection through these metabolomics. Here are the demographics of the study participants. So we have their age, we have their menopausal status, we have cancer stage, 1, 2, and 3, cancer types, so ductal, lobular, or both. We have their ER, PR, and HER2 status. We know which ones are triple negative. And then we have molecular subtypes too, based upon, of course, the histology results reported above. So what we did here, which was really interesting, and one of the first studies to do this, is that we had two control groups from two different locations, two different biobanks. One was from the Fred Hutchinson Cancer Research Center, and the second group of control samples was purchased from BloodWorks Northwest. So what we detected, so we ran this big screen. We had 105 metabolites that were detected in our quality control sample with a signal-to-noise ratio of 3. 98% of those metabolites were detected in about 90% of all samples and had a coefficient of variation less than 25% in QC samples. And then 30 QC normalized metabolites were not significantly different between the two healthy control groups. So of all of the metabolites we monitored, only 30 of them were not significantly different between the control groups. Only 30 of them, okay? So we monitored 105 metabolites, right? 75 of them had statistically significant differences between these two groups of healthy controls simply because they were from different biobanks. And we don't know why. So we continued the analysis with just these 30 metabolites that were not significantly different. Because if we're trying to then compare the control group to cancer individuals, how are we going to get a good comparison if they were significantly different between the controls themselves? So this is how we correct for geographic variation that is inherent in multi-site studies. So what we did was we took those 30 metabolites and we modeled them. On the left-hand side, you'll see a partial least squares discriminant analysis model, and then you'll see a statistical validation on the right-hand side. So what we're seeing here is that the model is not overfit. This Q statistic is, we're seeing is two separation, right? So based on these 30 metabolites, the ones that weren't significantly different between the two control groups, we can separate in the lower quadrant 4 and between this cross-section in the origin, we can separate breast cancer from control pretty clearly, okay? When we validate that model, so we're looking for model fit, we want to make sure the model is not too overfit and it can accommodate new samples, because the validity of a model is if it can be applied to non-trained cases. We see that the model is not overfit, so that's good. And we see here that we have very good predictive capacity and pretty good explanatory capacity as well. Relatively good explanatory capacity. So we're predicting cases about 70% and we're explaining them about 41%. So here are the 30 metabolites that were significant or non-significant between controls. And these were so these are the 30 metabolites that were not significant between controls, and these were the 6 metabolites that had all significant values. So the p-values are significant between breast cancer and controls. Their FDR, their false discovery rate, Q value. So when we do metabolomics and we compare hundreds of metabolites, we know that a simple t-test is going to come up false positive 1 out of every 20 comparisons. So if we're making 30 comparisons, hundreds of comparisons, and in a study you'll see thousands of comparisons, we need to control for that number of comparisons. So FDR is calculated as the p-value divided by the number of comparisons you're making. And then we look at the fold change, right? We were talking a little bit earlier, Gail, about a physician who saw some succinic acid and they were like, oh, this is definitely cancer. Well, no, we want to see what was the fold change, and was that fold change at a magnitude and direction consistent with the literature. So for 5 of these 6 metabolites, they were decreased in the breast cancer group. So the control had higher levels. In one of these metabolites, hypoxanthine, it was increased. Okay. Now we picked these 6 to model because when we look at the PLSDA model, there's a statistic known as variable importance in projection, or VIP. This statistic tells us how important that metabolite is to the model's predictive capacity. So if it's above 1, it's above average predictor. If it's below 1, it's below average, okay? So we picked these 6 to continue our modeling with. And here are their levels, relative abundances and standard errors for these 3. So we see that, again, in 5 of the 6, the breast cancer group is diminished. And here we see their relative abundance. For something like 2-hydroxybenzoic acid, it's near depleted. For hypoxanthine, we see this increase. Remember, this had a 1.6-fold increase. And these are all normalized. They're normalized to QC values, so any changes in system performance are accounted for. And all of these metabolites are significant at the raw p-value of 0.001, highly significant. When we take that model and we say, okay, you have levels of all of these metabolites, we have 6 metabolites, and we also want to factor in age. Age is a good predictor here. So we factored in age, and we see that it has an accuracy of 88.9% with a confidence interval of anywhere between 85 to 93%. So it can be anywhere as accurate as 85 to 93%. The sensitivity is 80%, the specificity is about 75%, and this model is the outcome of the PLSDA model. So it's literally saying coefficient metabolite number 1 plus coefficient metabolite number 2, onward, 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 plus coefficient age plus error term. And the error term is a combination of true error and some proportion of variance that we were unable to explain. And now we're looking at pathway analyses. So now we step away from the metabolites and we say, what's happening at the pathway level? We see 3 significant pathway effects with a high level of impact. Some of these pathways have a high impact, but they're not significant. Some of these pathways are significant, but they have a very low impact, low degree of change. So we're looking at arginine and proline metabolism, inositol phosphate metabolism, fatty acid biosynthesis. In the paper, we postulate that the degradation of arginine and proline is a very well-characterized immune escape mechanism, whereby this nascent tumor is camouflaging itself from things like natural killer cells, which would usually come along and eat it up. And by kicking off byproducts of arginine and proline, it's actually creating camouflage. And we postulate some pathways there. And we also saw some fatty acid biosynthesis, which was diminished. We actually postulated this was because it's not the biosynthesis that was diminished, but the degradation that was increased, which is a cancer hallmark for increased energy generation by this tumor. We're also able to now look at altered mechanisms of arginine and proline metabolism. So we have the metabolism up top, and this is done with a Kiagen software. And we're like, okay, so here's the arginine and proline metabolism. What are the underlying factors in that metabolic pathway that are being affected? So one thing that we saw was proline degradation. This was the immune escape mechanism. In addition to that, we saw the super pathway of citrulline metabolism, citrulline biosynthesis, proline biosynthesis 1, arginase 2 pathway, and proline biosynthesis from arginine. So this explains the arginine and proline metabolism alterations that we saw. We also saw significant deregulation in purine nucleotide degradation, purine ribonucleoside degradation, so changes in ribose 1-phosphate, guanosine nucleotide degradation in the third pathway, and xanthine and xanthosine. And so we're now postulating we see effects in arginine and proline metabolism, but what factors are underlying that, okay? So now we need a factor analysis. So factor analysis is, of course, this multivariate data reduction. We are trying to, just like you saw on the digital twinning graphs, right? We're trying to explain an outcome, like we did with the scores, with underlying factors, those 5 equations that acted as nodes, sort of gatekeeping and moderating the projections into 6 months. We're doing this with our data. So we took, we did this with CIFA version 2 software. This is really old software no one uses anymore. And what we're trying to do, and basically the matrix notation is this for factor analysis. It's matrix algebra. So what we did here was we create eigenvalues for all of our metabolites. We had 30, remember? 30 that were not significant between the control groups. And we're plotting their eigenvalues. And the eigenvalues show how much variance they explain, okay? So we saw here that, so there are eigens. So from our actual data is in blue. So we take our actual data, we plot the eigenvalues from the factor analysis. We do factor analysis, get the eigenvalues. Eigenvalues are a relative, sort of gauge of importance in the model. And then we do the same thing for our parallel analysis. So how many factors do you retain? A lot of people do what's called the elbow method, where they'll look for the elbow in this graph and they'll pick the factor right before, so that would give us 1 factor. But what we are looking at is parallel analysis. So we take a data set with the same number of people, same number of variables, 30, so 30 columns and however many rows we had, and then we permute that data. So we create totally random data, junk data, intentionally. And then we do factor analysis on the junk data and we plot its eigenvalue. The part where the eigenvalue of your data is less than the random junk data, that's how you know those are the number of factors you need to extract. So what we see here is that at the number 4 point, suddenly the parallel analysis has a higher eigenvalue than what we got from our actual experimental data. That tells us that if we were to extract 4 factors, there's no guarantee that the 4th factor we extract is better or worse than the random data. So we extracted 3. And then we put their factor loadings here, and we're trying to look for patterns. Higher loadings grouped together tell us something. What we see here is proline, glycoxamine, agmatine have very high factor loadings. We also see non-adenoic acid, palmitic, and stearic acid have high factor loadings here. And in the middle, we see indole, 3-indole propionic acid, and indole 3-acetic acid. So of those 30 metabolites, they're grouping as we see. Now we've determined the number of factors from our parallel analysis, and we interpret this based on our own data and our own interpretation, our own knowledge. So we say proline, glycoxamine, agmatine. This is arginine proline metabolism that we saw. The non-adenoic palmitic and stearic acid, that's fatty acid biosynthesis. And these middle 3 indoles, this is tryptophan metabolism. So our pathway analysis was able to say, okay, we see issues in arginine and proline, and we see issues in fatty acid biosynthesis. Our factor analysis added tryptophan to it. We were able to uncover some more patterns that we didn't have before. So let's put it together. So here we have arginine and proline metabolism. You see control, and then stage 1, 2, and 3. This is connected to tryptophan metabolism. We see tryptophan. So glutamate was normal levels, but downstream and upstream we see dysregulations in glutamine. So that right? So we see agmatine, which is a precursor, glycoxamine, and proline. These are affected, but glutamate is not. Same thing with tryptophan, unaffected, but indole 3-acetic acid, indole are affected. For fatty acid biosynthesis, between the stages, we only had significance in palmitic acid. So this is our fatty acid biosynthesis. And we also see central carbon metabolism represented here, and glycolysis and gluconeogenesis. No differences in lactate, no differences in glucose. Krebs cycle, same thing. No differences in alpha-ketoglutarate, succinate, or fumarate. So we're able to map these. This is before we had AI. So these graphs look like this is in 2019. I was a grad student, right? So now we use AI. Okay, so the next thing I want to talk about is this new method we detected. So we were like, okay, we have a targeted panel we ran on these people. We only ran 126. What if, or not 126, I think this was like 330 with LCMS. I'm confusing with our own panel. What if we could develop something that didn't need internal standards and could use online analysis with databases? What if we could use the databases in real time to increase what this mass spec is able to see? So I came up with this paper. It's published in, of course, the very prestigious Analytical Chemistry, which is like the flag bearer for the field. And we termed it DGOT-MS. This was a period of time where Game of Thrones was incredibly popular. We were all fans of it, and we wanted to create an acronym that was in some ways paying homage to that. So yeah, Alex was talking about the acronyms that we like in science, so we went with DGOT. Here is the workflow. So we start with HMDB and Metlin. These are the two big metabolomics databases. They have predicted spectra for all of these metabolites. Then we look at the MRM transition. So here's the base peak. Here's the satellite peak after we break that base peak up twice using MSMS. We optimize their positive and negative mode. In our mass spec, we only use positive ionization. With LCMS, you can use negative ionization. Positive is for nitrogen-containing compounds, and negative is usually for all the rest. And we can optimize the collision energy. We don't have that in our mass spec because it's a single quadrupole. We have one quadrupole that is filtering all these metabolites. But in an MSMS, you have two quadrupoles back to back. They call it triple Q, but the middle Q is actually a nitrogen stream that breaks apart the molecule. So the collision energy of that nitrogen is going to be set. By optimizing these two, we're going to get optimized MRM transitions, so better peaks, well resolved. And then we're going to create this internal database from HMDB and Metlin that is going to run in real time. As the mass spec is collecting data, it's going to assign metabolite names, MRM transitions, and it's also going to look at collision energies, retention times, and CAV, which is another boring mass spec parameter. And then it's going to do this dynamic MRM, so it's going to know what the database is telling it should see based upon the chemical signature, and in real time, it's going to be able to identify 9,000 metabolites at once. There are 25,000 metabolites in the HMDB. We cannot afford to buy standards for them, and oftentimes standards aren't available for some of the most relevant compounds. But if we can utilize this knowledge that's deposited online, bring it into an internal database that we curated for months by hand, and then connect that with the machine that's collecting this data, in real time, it's going to be able to identify 9 of those 25,000 metabolites. 9,000, sorry, of those 25,000 metabolites. So what we have here is tryptophan, right? So here's some of the formulas and everything else. Here's the chemical structure. We know that it has a nitrogen, so we know this is a positive mode data collection. Here's the HMDB spectra that we should see, okay? And then we're optimizing the collision energy. So this is the spectra we should see. We optimized it at 3. We chose 3 different, usually commonly selected collision energies. Those are going to be at 10 volts, 25, and 40 volts. And we saw that for this instance, we saw better product ions from at 10 volts, but at 25 volts, we saw satellite peaks that were very detectable here, and at 40 volts, we saw a satellite peak that was shifted slightly to the right and decrease in intensity. So when I talk about, well, how much can we see? So this was scanning from 50 to 30, 350 mass to charge. We scan from 60 to 500. And this is how the data is collated, right? We have a retention time. You guys know what that is now. And we have a mass to charge, right? If we were only to look at the mass to charge, we would see all these retention times stacked against each other. It would be a very messy thing. So we need to go, okay, at 5 minutes, what is this peak here? So we were able to, in total, look at, I think, 9,058 different metabolites all at once. And we'll talk about the confidence with which we detected them, right? So these are the ones that were identified with very, very, very low CV, coefficient of variation. CV is standard deviation divided by mean. So it is a ratio of how much deviation per unit of average you're seeing. You want this very, very low, ideally below 20%. That's the analytical rule. And we have intraday CV, DMRM intraday, DMRM interday, and linearity. So we see incredible agreement in 3 different separation phases. We had acidic reverse phase for lipid compounds. We had acidic helic, and this is hydrophobic interaction liquid chromatography. So these are for the aqueous, these are for the lipids. And what we're seeing here is different levels of identification. And we're seeing them at intraday CV of 11%, 4.8%, and then we also see 8% here with a linearity, meaning that what the database predicted we should see and what we saw was 96% in agreement. So the method works. So we then graphed identification levels 2 through 4. Level 1 was everything we had an internal standard for. Of course, we're going to identify that one. But what we're really interested in is level 2, where the metabolite is confirmed by at least 2 MRMs, 2 satellite peaks following collision. And then if multiple peaks were produced, we chose the highest one. Level 3 is where we still have more than 2 peaks, but if the multiple peaks are produced, the heights are basically like the same and we can't distinguish between them. That's 8% of the total metabolites we detected. And then level 4, and these are all metabolites that are identified with good coefficient of variation, is where we only have 1 MRM. So we go in decreasing order of confidence, but we can see that most of the metabolites that we detected were of very high confidence and had good annotation. When we look at the distribution of these precursors and product ions, right, we see the precursor on the x-axis and the product on the y-axis. So we know that when we have a precursor of 800, you should probably see a product of about 400. And what we see here between a traditional targeted in the positive ionization mode and our DGOT MS is significantly more coverage here, especially in the range between 2 to 600. We see much more density. Same thing here. Each one represents a compound that we can look at with low CV and high confidence. Same thing here in the negative mode. Positive mode is a little bit more obvious because positive is related to nitrogen-containing compounds, but we see significantly more density there as well. So we look at the intraday coefficients, right? So how many of them, right? We see intraday DMRMs, intraday, and then intraday MRMs. And the percent of peaks are really high in the intraday MRMs, and the median coefficient of variation is significantly lower than clinical guidelines of 20%. In fact, for studies, they'll do a CV of 30%, which is just absolutely ridiculous if you ask me. How can you have a CV of 30% in the quality control, and then you have a significance level of 5%? This is ridiculous. So in our studies, we usually favor very, very low significances to overcome some of these differences in variation. So very, very accurate and reliable method. Now, what happens if we take our traditional method, right? We use, like 330 metabolites, or what if we go to this like 9,000 panel? How much separation do we get? We do see significantly different and significantly improved separation, whereby we don't see as much dispersion, specifically in the control group. In the DGOT group, we see a good delineation here, whereas with the traditional targeted, we do have some, these are individuals, these are samples, not metabolites. So these samples are crossing over into the confidence interval of the BC group. These are breast cancer patients. So we had some breast cancer cell lines. We thought, okay, we developed this method. We proved it's analytically valid. Let's apply it to breast cancer, right? So now we're looking at markers of breast cancer. What we see with the traditional targeted method, and this is a volcano plot where the fold change is set to more than 1.5, so at least a 50% increase or 50% decrease, and the significance is set at 0.05. This is how many biomarkers you would return with the traditional method, like the one we used in the first study. This is how many we're now able to return reliable, statistically significant biomarkers with this DGOT method. That's it. Now we're going to go to early breast cancer detection. Again, here is the table of demographic and clinical characteristics. We have their age, cancer stage, cancer type. We were not able to accurately look at stage 1 cancer in our previous study. This is kind of where we come back and we're like, okay, let's see what we can do with our new method. Can we detect stage 1 cancer? So we ran our DGOT method. This is on liquid chromatography, quadrupole time of flight mass spec. And what we saw was between stage 1 and control, stage 2, stages 1 and 2, and stage 3 and control, these are all the comparisons. These are the significant metabolites between those. So sometimes a metabolite was significant in stage 1, dormant and non-significant in stage 2, significant again at stage 3. This happens. This happens as cancers progress and metabolic pathways shift. Things that were once significantly different between their diseased individuals and their healthy counterparts no longer are significant.
So would you say that happens more often than not?
No. It's very nuanced, but it's important because somebody will say, oh, you know, my succinic acid levels were elevated. I thought I had stage 1 cancer, but at retest, they're really good. Are they really good, or are you preparing for one of these to come up, right? Same thing here, whereas, you know, you have like allantoin, right? Not significant, or not allantoin, what's this one? Lyso-PE, right? Or biliverdin, both of these. They're not significant at stage 1, they're significant at stage 2, and then not significant at stage 3. They just go away, right? So this is what we were able to detect, and as you can see, we're able to get a whole splattering of things. Some of these are unidentified. Master charge 586, we don't know what it is. It just has a master charge, right? So this method is untargeted. The first paper was targeted. We're looking specifically at these things that we have standards for. But what if we were to step back and not look at anything specific and use these databases in real time to inform collection on 9,000 metabolites? So we have these. Some of these are, again, master charge 261. Some of these are lipids. These are not easy to detect, especially in the same run as aqueous metabolites, since they're chemically different. So here are the 6 biomarkers that we really saw. And these are the ones that are very predictive between early breast cancer and the control groups. 3 of these 6 unidentified mass to charge ratios. They serve us absolutely 0% when we talk about biological interpretation because we don't know what they are. However, as reliable predictive markers of cancer, they're quite accurate. Remember our first study, hypoxanthine, was elevated? The only marker that was elevated, it popped up in this study as well as elevated. Separate study, totally different platform. We did models here. So control and early breast cancer, we did a trial on the cell lines. You saw separation in the method previously. Now we're looking at the same subjects from that first study. These are the same samples that we ran again on this new method. So we published, we saw we couldn't get early breast cancer. We developed a really badass method. We went back to the same samples after we proved them in breast cancer cell lines, and now we see really good separation, especially with this increased sample size. We see good model permutation. Here we see the predictive capacity of the model is, I don't know, how much is it, like 90% or something. What is that one? See, external, 86.8% with 50% of the samples as holdout. And then we do a training set, and that's the straight line. You can see that here. This is iteration, so this is internal statistic permutation, and this is the training set. So the training set actually, this is the training set with the internal statistical permutation. This is our testing set. And in the testing set, the model actually worked better than it did in the training set. We had 93.8% classification with just those 6 markers, not age included either. 3 of them were unidentified. We had no idea what they were. So it doesn't help us in terms of like biological interpretation, but we can set this machine up again and say, look for these master charges. Their differences will tell you if it's early breast cancer. And then this is the optimal cutoff line. So these are model. So what we can see here is that early breast cancer has very little variation. They're all stacked here. Con has a little bit more variation. We try to find the optimal cutoff line that maximizes this area under curve, maximizes the accuracy. This is known as the Uden method. And then we can start looking at the metabolites that were identified with actual identification, not master charges, and start mapping them. What do we see? Again, we see glycerophospholipid metabolism. We see issues in fatty acid metabolism. We see issues in arginine and proline metabolism. We see issues in glutamate and glutamine metabolism, and some others that weren't detected because now we have a broader panel. But we see the results of our first study echoed really well. Same samples from the same biobank. All right, now we're going to look at this other study we published. This is me and a professor up on the third floor. She's the director of the translational team and invited me on to do some metabolomics and statistics. This guy was like one of my heroes. He's like one of the founding fathers of metabolomics, so I only agreed because I got to work with him. This is we have two trials, one in Italy, one in the US. Randomized, this is the Reach for Health study and the MET-BRIE-CS study. These are the different conditions of each of these groups. So we had placebo control, metformin control, weight loss, placebo, weight loss, metformin, placebo, metformin. And then we had this second study, which is we're looking at the main effect of metformin in groups for the metabolomics. So we basically took the results of all of these and we just classified them. We mixed them together and we classified them between placebo and those receiving metformin. These are breast cancer survivors. Yeah, breast cancer survivors. So these are the trial characteristics. We had a ton of really important data on them for both the Italian and American cohorts. Weight loss percentage, age, BMI, menopause status, pathological stage of their cancer, histology, tumor grade, ER, PR, HER2, and if they were on aromatase inhibitor therapy. What did we find? Well, we found that people who were taking, so here we're looking at these Spearman-based changes. In the A and C, you'll notice these are volcano plots. This is, again, looking at full change in significance. These are the two things we look at. So when people say, like, again, the physician, Greg, who is about succinic acid, it's not just that it's above the range that we report. The range that we report are normal physiological ranges in people with no evidence of disease. It doesn't mean that if it's higher or lower, it's cancer. Right? It has to match with the significance and the full change that's reported in the studies that are powering that. So you'll notice in the disease signature report, breast cancer or something might pop up. There's a column that says total monitored metabolites and then matched metabolites. We all have those, 54 metabolites related to breast cancer or whatever. It's what proportion of those 54 were expressed with a magnitude of change in a direction that is consistent with breast cancer. So here we're basically looking at pathways that were responsive to this metformin therapy and actually led to a significant decrease. So those who were in these trials and were receiving metformin, they actually had far lower levels of cancer recidivism when they were being tracked. And we are looking at metabolites and specifically pathways. These are the master charges, right? So this is 232 at 15 minutes. This is master charge 131 at 0.93 minutes. 192 at 21 minutes, and we're looking at identifying patterns in these heat maps that explain why metformin is working for these people. So we're postulating effects, related to 3 and 4 methyl oxyvalerate, isoleucine metabolism, and indoxyl sulfate metabolism. Caffeine had a role to play in this. It's something that we monitor in paraxanthine as well. So here are the level 1 identified metabolites. So these are all confirmed with internal standards with the metformin treated groups and how they change significantly or differ significantly from the placebo over time. And they were given placebo. So some were given, obviously when you give someone a pill in a randomized controlled trial, you have to give the control group something to correct for the placebo effect. And what we see is that caffeine goes down along with paraxanthine and theophylline, and then we see significant increases in the other metabolites. And these are the coefficient of variation in that sample. As I mentioned, this isn't my data collection, so I would have kind of scoffed at this 24.14%. Analytically, it should be less than 20. And so it induces these really broad changes, right? Here's the beta regression coefficient. So if we were to create equations of these to predict response to metformin, we would see that the most important is, negatively important is citrulline, and the most positively important is alanine. And these are their FDR-corrected p-values. So these are the stacked plots of various fatty acids that were really interesting. We also saw some changes in odd-chain fatty acids. They were borderline significant, but it was really interesting because odd-chain fatty acids are gut-derived. We don't make odd-chain fatty acids. There's a 15-carbon fatty acid. We don't create that, so it's of microbial origin. But these are not, and this is really just looking at how metformin affected fatty acid profiles over time. And this was one of the postulated mechanisms by which we inferred that metformin's actions for breast cancer were relevant. And this is a network analysis where we're just looking at pathways and positive correlations. The nodes, of course, if they're red, they denote negative correlations, for instance, between BMI and this phosphatidylcholine node. These are the parameters underlying the models that we are using to predict. So oftentimes we will use demographic data. We'll use BMI. We know that women with higher BMI are more susceptible to cancer. We know that smoking is one thing that's going to lead to increased breast cancer in individuals. So these are the network. I mean, Alex alluded to this. There's really nothing that you need to really concern yourself with in this report, but just know that disease is not, especially when we talk about disease identification, it's no longer at the biomarker level. It is at the network-based pathway level. And these are where you see effects. So if you want to personalize something, you look at the pathway. You don't just focus on one marker, because as we saw with the breast cancer stages, something will be significant, then it won't be significant, then it will be significant again. You need to look at entire pathways because those don't lie. Okay, and that's it. You're free. Any questions? No.
Do you have the child book version of that with the pictures?
They pop up and... Yeah. Well, we started pretty shallow. First talk was just like, hey, welcome. And then the second talk was like, we're getting into testing. And then the third one was like, okay, let's open the hood and see what actually powers these things. So you saw everything from how do we create a model? How do we then create a method to improve our detection? How do we apply that method to cells and then humans to look for early breast cancer, which we weren't able to see? And then what kind of treatments do we give for this individual whose cancer is in remission? How do we prevent recidivism? And how do we then take those information, the dosages that they were taking, we can model that for our individuals. We do the same thing with niacinamide or Bacopa Monnieri or lion's mane. There are clinical trials on these supplements, and we can map the dosages to their metabolite effects. So in that instance of that individual who we were like, okay, we want you to take your phosphatidylserine, same dosage, break it up into 3 smaller dosages throughout the day. There's a reason for that. It comes from studies like this who, they've taken years of their life to show effects, map effects, and enable us to model it with the data they publish.
A lot of the challenges I feel that testing companies have is validation. And so what you presented, I think, is a really detailed and robust way of trying to validate what you're trying to test for.
Yeah, absolutely, Michelle. Yeah.
So is it mainly they were saying that metformin and women that have had breast cancer, does it protect them from getting it again?
Yeah, absolutely. So I didn't show some of the other stuff, but we tracked them through time. Yeah.
I mean, of course, metformin's to lower blood sugar.
Yeah. Yeah, we saw that. So we had things like weight change. We had BMI. Like those were in the clinical characteristics because those are the effects of metformin. You need to track those and correct for them. And we can do those things. Like, right, we can put models, we can put in variables as covariates. So they account for things, right? For instance, when we did the screen time paper, a relevant covariate is physical activity. So we include that so the model accounts for physical activity, corrects for it, and so we can be sure that the biomarkers we're seeing are from the variable of interest and not from some confounding variable that's mediating or moderating that relationship. When we look at aging, I have a paper on aging, and one of the most difficult things to do is how are you sure the biomarkers you're getting are biomarkers of aging and not of disease? Disease comes with age. So you do, some calculated stuff. You do like chronic condition score can be calculated and included in that model as a covariate so that you can be sure that the signal you're getting from the biomarkers is corrected for any chronic conditions this person may be having. And the signal, the response you're getting from that biomarker is, again, going to be reliable indicator of aging and not confounded by disease processes that are associated with aging, right? So there's a lot of work that goes on to this, and then I get on, and then I love the physicians. Well, we'll get on a consulting call, and they'll be like, "How do you know this?" And I'm like, "Well, I mean, I can look up the database and then look up the studies and see how they were validated and how we're using them, and that's going to take me 5 hours, right?" And really, this is kind of opening up the hood, opening up the digital, huh?
You need to watch their movie,
Hidden
Hidden Numbers.
Numbers. Yeah, why?
Well, because
Just because it's entertaining, or?
no, because that is exactly the same problem. Is they don't understand, and they ask you. The question is, well, how you know this means I didn't understand anything what you do.
Yeah.
So now you are trying to convince somebody who doesn't know your lingo.
Right. I mean, we had a physician reach out, and
It's impossible.
it's impossible. Yeah.
It's impossible task.
And isn't the main problem there, they just weren't trained in that area?
And we don't expect you to be. Like, we don't, like,
But then you will not be able to sell it.
well, that's where you come in as the human shield. And, and you go forward and you say, hey, I'm one of you, and I trust this guy, and go trust him, right? So we really have to lean into the physician thing, because with this physician, and then she said, oh no, succinate is an indicator of this. And then she sent like 2 of these links, and they were like awful studies, like awful design, execution, analysis. And she's sure of it because these 2 studies, just because it's on PubMed, does not mean anything. Everything gets scraped on PubMed. Everything gets put on PubMed, right? I could write a letter to my mother this afternoon. By tonight, it's published on PubMed. It's meaningless. It's not
The problem is that you know what, again, the question or problem you're facing is that you read the papers. Now you understand what it's supposed to do, and then you read numbers like this doesn't make sense, like this 24 number, and you know this. The
Right.
standard is 20.
Yeah.
But many people don't know. They mean somebody already sanitized this, and they believe that what is on a PubMed is already sanitized and it's
peer reviewed.
It's peer reviewed.
It's broken. Peer review is broken, by the way. You've seen the studies where they've put in
I understand.
crap papers and you've seen the comments back.
Now you have, like you see now on TV, that they start talking about board director of New England Journal of Medicine says 50% what we publish cannot be really considered as a truth because it's just crappy.
10,000 papers retracted last year.
So that means that a lot of people will now don't believe anything because they are like, yes or no.
Yeah.
You have advantage that you understand all, it's like, oh, this cannot be true because, and you know the lingo. Many don't. They are forced to make decision today.
Yeah.
And she believes that this, that's exactly what oral micro, I use that example.
Yeah. Yeah.
So that bridge, these translators are extremely important because you cannot convince the doctor who doesn't understand all this.
Well,
Like you
the
have-
other factor is replication. We're continually showing. these
Yeah, with hypoxanthine in 2 of our own studies. 2 different methods, same samples, showing hypoxanthine in a very similar fold change. That is telling us that this is a reliable indicator. We also saw like xanthine also changed as well. So metabolites related to hypoxanthine are affected. So you can be relatively certain of that. So that's where,
Same like in the study you said, sometimes it's not relevant. Then the first stage, then nothing in the second. The third, it comes up.
comes up again. Yeah.
Well, but somebody will say like, what am I supposed to tell a patient?
Well, hopefully we can tell you what to tell the patient.
Well, that's exactly, that's the challenge.
And we have to build trust for you to be able to then do that. Yeah.